So I will speak to you a bit about our modeling efforts that we have been doing in Dublin. Specifically, we've developed a theoretical model for how to describe nanoparticle accumulation in dividing cell populations. So the reason for doing this work in the first place is that, of course, there are many people who are doing in vivo tests and working with primary cells, but I think it's safe to say that most studies on nanoparticle uptake and toxicity, etc., are performed on cell lines. And in cell lines, cells divide. And when the cell divides, whatever nanoparticles that are inside the cell, they will be split between the daughter cells. And that is something, regardless if you're interested in looking at nanoparticle accumulation or toxicity, is something that needs to be taken into account. So this talk will be about a framework that indeed does take this into account. So on your right here, you will have essentially a cell cycle model. I will not go into the details of the mathematics here, but the basic point is that it's a rather simple model, at least from a mathematical point of view. And in fact, the expert listener might recognize this as a wave equation. So essentially, we see cell cycle progression as kind of like a wave of cells going around the cell cycle. The nice thing with this kind of model is that if you look for time-independent solutions, so that's well, actually it's where the fraction of cells is time-independent. What immediately materializes from this kind of model is this exponential growth here, up there. So that's very nice. This very simple model, immediately we can identify solutions to the model with exponential or asynchronous growth. And the second part of this solution that you can see here gives us how, let's call it the age distribution of cells. So that is how many cells have just divided, how many cells are in G1 phase, how many cells are in S phase, how many cells are just about to divide. And the very nice thing is that these kind of parameters are actually experimentally measurable as well. So from experiments, we can measure the number of cells in G1 phase, in S phase, and in G2M phase, and then put that into the model. And having done that, then there are no free parameters left. So we have, this is for A509 cells, but I guess the details wouldn't matter much. It would just be different parameters for other cells. So having done that, we have a model that is for A509 cells, and we can use that model. So experimentally, one can measure a certain subpopulation of cells and see how many cells divide as a function of time. And if you can do exactly the same thing, again, I stress that this is a parameter-free prediction. It's not a fit. And you can do the same thing from the theory, and you see that they overlap rather nicely. So in this way, we feel that we have validated at least the cell cycle part of the model. So going further, we just introduce a second parameter to this distribution function that measures the number of nanoparticles in each cell. Again, I don't want to discuss too much of the mathematics, but I would. One important thing is the flux here, so we assume, or depending on the experiment, but if we have an experiment where particles are continuously taken up, we assume that there is a given flux into cells. I might also note this function that I call theta in this particular case, which is about how nanoparticles are distributed among cells when the cell divides. And that's something that the Hugh Summers and the Swansea group have studied quite a lot, but he actually didn't speak about it this morning, so that placeholder was kind of gone. So having this kind of framework, we can also start using it for actually predicting stuff. So one thing we have been rather interested in Dublin is the question if nanoparticles, once they're taken up by cells, do they ever go out again? And we have developed methods for testing this experimentally, as you can look at, you can find in the first paper there. Where you can also use the model to strengthen these kind of things. So on your left here, that's those are cells that have been exposed to particles for 4 hours, and then the particles are washed away, and the cells are just left as they are. And we measured the number of nanoparticles per cell as a function of time. And what you can see there is an exponential decay, and the exponential decay fits very well to the cell population doubling time of these particular cells. Now, of course, you don't need a complicated model to understand that would be an exponential decay, but the nice thing is the graph on the right there, which again is a parameter-free prediction. So having done the fit on the left side, we can just take the model and run it and see what it gives. In this case, for two particular subpopulations of cells, and as you can see, the data points there, here and there, they are exactly on top of the prediction from the model. So we're fairly happy with that. So essentially now we have a model for nanoparticle accumulation and cell cycle, and we can use it to essentially whatever you can measure experimentally, we can also calculate it from theory. So another example here is the nanoparticle accumulation of cells during continuous uptake. So the previous graph was for like pulse loading or whatever you want to call it. This is with a continuous source of nanoparticles. What you tend to see, or at least what we tend to see, is an initial linear uptake here, and then kind of a saturation as time goes on. And one reason for such a saturation phenomenon could be that particles are exported, but we've just shown that particles are actually not exported. And there could also be a range of other behaviors, but we find that we can actually fit this data rather well to this model with cell division. So essentially what happens is that nanoparticles are continuously taken up, but as the particles are taken up, the cells are also dividing. So you have two competing processes, and eventually they reach a plateau and eventually an equilibrium, even though that hasn't happened yet in that data. As an added bonus, we can also, from the model, it also comes out exactly what that kind of flux that is in that equation actually is. And it's essentially a weighted average of the fluxes into the different phases. And, well, this is just another example, continuing calculating even more things from the model, things that we have also measured experimentally. So you can also look at the number of nanoparticles of cells that are in that moment in a particular phase. So in this case, G0, G1, S, and G2M. And if you do the same kind of calculation from the model, you again get rather good agreement, both at the mean population level that you have here, but even going into the actual distribution. So the black line here would be the full population, whereas those other colors would be individual cell cycle phases. So we believe this model works rather well. Now, for the last part of my talk, I just want to speak a bit about what this flux into cells actually is, and well, at least in particular, what does it depend upon. So the first thing that maybe by now is rather well recognized is that this flux into cells is most definitely energy dependent, meaning the cell is actually spending energy to take up the nanoparticles. As you can see here, where you have cells taking up nanoparticles and other cells that have just been exposed at 4 degrees or with sodium iodide, and they don't take up any nanoparticles at all. Of course, I can't fail to mention that another reason for another thing that affects the uptake rate is the biomolecular corona. And I think this graph here on the right is very illustrates it very clearly. So this is the uptake in the absence of a corona, so a bare particle. This here, the lower one, is the uptake with the corona. So there's actually a huge difference in uptake if you have a corona or not. And as you can imagine, you will also have a huge you can have completely different impacts from nanoparticles as well. So in fact, in this particular case of silicon nanoparticles, without a corona, you have the cells start dying, and you also see nanoparticles free in the cytosol. Now to go a bit further and actually understand this uptake rate, we also started studying how the nanoparticles are adsorbing to the cell membrane. And again, this is a process that is very much dependent on the absence or presence of a protein corona. So if you look at the upper one here, you should compare that to the lower graph here. That's the same nanoparticle concentration, but with and without protein. So you see that without protein, the particles stick to the membrane like crazy, essentially. But as soon as you put something on top of them, they still do stick, but they stick a lot less. And this is just to demonstrate as well that, in fact, they actually do stick. You see a big difference here. It's just that in comparison to without a corona, you don't see it very much. So to model this, we have just developed a very simple model where we describe the uptake of nanoparticles as first the particle is finding a cell and sticking to it, and once it's absorbed to it, you have to wait a bit longer until it's actually taken up. And you can see that if you write up this kind of equation system here, and you turn out to look at the nanoparticle flux, which we have measured experimentally, you again get a rather good fit with experimental data. So, to summarize, the reason for doing this at all is that typically most experiments are performed in vitro on the cell lines, and the cell lines cells divide. So you have to take this into account when you do experiments with nanoparticles. We have developed a simple framework for this, and we have validated it rather extensively, both with parameter-free predictions, as in this case, and with fits to data. So we think it's a rather sturdy framework. And this framework can then be used to calculate essentially everything you can measure experimentally. The flux that is included in this kind of framework, it most definitely depends on the cell energy. It also depends very strongly on the presence or absence of a biomolecular corona, and we can describe it in terms of an adsorption and then internalization into the cell model. And that's what I want to say today. I should acknowledge my experimental colleagues, Anna Salvati, Shona King, and Anna Leslie.
Thank you, Christopher. We're now ready to take some questions. Yep.
Thank you, Christopher. I'm more confused now than before you talk. No, excellent. Thank you again. But what I thought is that silicon nanoparticles, like very small nanoparticles that don't have any specific coatings, they enter through a non-specific mechanism into the cells. That's what I thought. There is no like a receptor or non-clathrin mediated uptake. So, but from your data, you are suggesting there is a specific uptake that is depending on the coating, or it's something that you haven't investigated?
Well, most of what I showed here was actually for polystyrene particles, and the few things I showed with silica was actually rather big. They were 50 nanometer. But regardless, everything we've seen, they enter via energy-dependent pathways. But I imagined if you go into very, very small sizes, there might be other effects, but that's not something we have really studied.
Thank you, Christopher. I would like to introduce our second